Good morning. My name is Rebecca Sharif, and I teach biology. Yesterday we were analyzing how food is formed by these glands, and we severally looked at the C3 and the C4 glands. I wanted to emphasize C4 glands. These glands here, they happen to be, they possess a tendency of overcoming the respiration, the photo respiration that is demonstrated. I want to remind you on the carbon dioxide acceptor. We said that it is the three carbonic compound that accepts carbon dioxide. It accepts carbon dioxide, and this enzyme PEP carboxylase. PEP carboxylase, it's a carbon dioxide enzyme that catalyzes the reaction between PEP and acceptance of the carbon dioxide. We say that the PEP has a very high affinity for carbon dioxide, so it accepts carbon dioxide. It accepts carbon dioxide and it will form a new substance. Normally say it is oxaloacetic acid. Oxaloacetic acid. Now, this is a 4 carbonic compound. Because we had a 3 carbonic compound, and we added another carbon from the carbon dioxide, so we formed oxaloacetic acid. Oxaloacetic acid will form malic acid, and now we shall have this malic acid, a 4 carbonic compound, forming pyruvic acid, generates pyruvic acid. So pyruvic acid, it's used for regeneration. It's used for regeneration of the PEP. So here, pyruvic acid regenerating PEP, it is an energy consuming process. So an ATP molecule will be broken down to ADP and inorganic phosphates. So you take note that we have 3 carbon compounds by the pyruvic acid, and now we are wondering why the 3 carbon atoms are presented here, and the malic acid here, it is demonstrating the 4 carbon atom. It is an indicator. That the carbon dioxide is released out of the reaction here. I want you to take note of that. Carbon dioxide is released out of this reaction, and this carbon dioxide, it goes to the C3 cycle. It goes to the C3 cycle. And now this carbon dioxide is going to be accepted by the Ruby, and the reaction will proceed like we mentioned in the C3 plants. So this is called C4 cycle. Something unique about the C4 cycle is that it occurs in separate cells. There is a division here which will show that PEP accepting carbon dioxide in the form of oxaloacetic acid. This will occur in the bundle. This will occur in the mesophyll cells. Mesophyll cells, and then this other separate malic acid forming pyruvic acid and regeneration of PEP. These two reactions happen in the bundle of sheath cells. I was emphasizing that they occur in different types of cells. So PEP accepts carbon dioxide in the form of oxaloacetic acid. This reaction happens to take place in the mesophyll cells, and the malic acid producing pyruvic acid and the fixation of carbon dioxide to the Kelvin cycle, or what we call the C3 cycle. It occurs in the bundle of sheath cells. You see, if you look at the structure of the leaf, you realize that we have the palisade mesophyll and the spordium mesophyll cells. This reaction will happen in the palisade mesophyll cells, and the same happens in the spordium mesophyll cells. However, the palisade mesophyll cell have a high dominance on how this reaction proceeds because it contains much of the chloroplasts. The bundle of Buschiti cells are those cells that are comprised of the xylem and the phloem. So the fixation of carbon dioxide actually occurs, and that is said to be a Kranz's anatomy. So ask you what is meant by the Kranz's anatomy. You'll be in position to identify that during the C4 cycle or the Hertzsprung pathway, fixation of carbon dioxide occur in separate cells to the fixation of carbon dioxide during the cycle. So we are fixing carbon dioxide in C4 plants twice. The first fixation occurs by the carbon dioxide acceptor pair, and you say this is influenced by an enzyme carboxylase. After this reaction, after the acceptance of this carbon dioxide, because the pair will have high affinity for carbon dioxide acceptance, we shall generate malic acid, and eventually carbon dioxide is fixed in the C3 cycle. I want to emphasize that on there. So I'm mentioning that the reaction will happen if you separate the mesophyll cells. That's why you have the first fixation of carbon dioxide, and the second fixation of carbon dioxide in the C3 cycle separately occur in bundle of Buschiti cells. I will demonstrate here. I will briefly demonstrate here on how the anatomy of the leaf would appear. You must have anatomical structure of a leaf to appreciate how these reactions occur. That is the internal structure of the leaf. The first lining cells are longitudinal. You can see them, and these ones here are said to be palisade mesophyll cells. They are present in the palisade mesophyll layer. These cells dominate with a number of chloroplasts. I want to demonstrate with one. By similar, even the other. Consider this to be the nucleus. Now you will appreciate that the chloroplasts are the functional structures in the cells of photosynthesis. So we want to appreciate that in the chloroplasts on the thylakoid membranes, like we mentioned earlier, we have the photolytic pigment C. So there are photolytic pigments here. Chlorophyll dominates, you are aware. So we expect these cells on the leaf to appear more green than the other cells, and that one will be demonstrated later. So we have the spongial mesophyll cells here. These cells we call them spongial mesophyll because they create a spongy, texture and appearance of this part of the leaf. They create air spaces because they are circular to allow circulation of gases around the leaf like that. Now, adjacent to them shall have the bundle of Buschiti cells. These involve the xylem and the phloem. So we have numerous xylem inside it. Now outside here, that's where we have the phloem. So these cells here, they are xylem. Probably this one outside is said to be the phloem. That these are spongial mesophyll cells. Palisade mesophyll cells. Down here we have the epidermal cells. These epidermal cells are running horizontal to the leaf, and of course in between them we severally have B-initiative cells. Normally define them as Guard cells. So the Guard cells generate an opening called the stoma. Here's stoma, right? And now that is the zero structure of the leaf. Of course, the palisade mesophyll layer, they have got a thin line of cells alongside them. And this one here, I think should be a cuticle. We can call it a shiny cuticle. And now the leaf, when you look at the thickness of the leaf, would have that structure generally. This is said to be the upper epidermis, or you can simply call it the upper layer. The upper layer of the leaf. That is the upper layer of the leaf. However, this one here, the lower layer of the leaf. You can say lower epidermis. Lower epidermis structure. So we have several spongiomesophyll cells here. You can draw very provided you show us that there are spaces created between them to allow circulation of air. Circulation of air, no air enters into the leaf passing through the stomata, and then it exits out of the same route. Say carbon dioxide can enter, and then carbon dioxide can be discharged out, oxygen can be discharged out. We mentioned that oxygen is produced during the light stage, you remember. And now carbon dioxide is utilized during the darker stage, you are aware. So we need to appreciate how these gases enter. Some other questions can be made here, and they can involve you in explaining the ventilation in the plant. You see the plants, for them, they don't have the lungs like human beings, but they have a specialized mechanism on how they allow air to reach those parts that require them, and then they can exit the gases produced from these living cells in the plant. There you will specify, talking a lot about the spongiomesophyll cells that are in the leaves, and also emphasizing that that's one of the reasons why the leaves are numerous, to ensure the increased surface area for this gaseous exchange. So a leaf is a plant organ which is greatly complicated in functioning. From the several of them, however, our attention is much more on the nutrition now. The palisade mesophyll layer happens to have chloroplast, numerous of them. If you are looked at a leaf and you look at its upper surface, any leaf, you realize that the upper surface of a leaf happens to be more green. And when you turn it around to the other side of the lower, you will appreciate that the green there is lighter. That is an indicator that the palisade mesophyll layer that is situated on the upper epidermis proves with the highest chloroplast concentration than the lower. So the lower epidermis, there is the chlorophyll there. We appreciate there are some chloroplasts, but there are very few on the lower epidermis. There are very few, if any. And the dominating in the upper epidermis. What is the adaptation? What is the significance for this positioning? All of you must be thinking about it already, that it maximizes the absorption of sunlight given that positioning. Maximizes the absorption of sunlight, and thereby light finds its way to the other regions. When you come here, on our illustration of the C4 plants, the C4 cycle, or the Hertzlanger pathway appreciated yesterday, I wanted so much to emphasize that carbon dioxide fixation to PEP, or PEP accepting carbon dioxide, it occurs in mesophyll cells. See? Maybe palisade mesophyll or spongy mesophyll, but more dominantly in the palisade mesophyll cells. But if it comes to formation of oxaloacetic acid and now malic acid generating pyruvic acid, carbon dioxide is fixed in the C3 cycle. This fixation of carbon dioxide in the C3 cycle, it occurs in separate cells other than in the mesophyll cells in that is there. So you realize that if PEP was in this particular cell accepting carbon dioxide which carrying, going to have PEP discharged and formed oxaloacetic acid. Oxaloacetic acid form malic acid in these other groups of cells of the xylem and the phloem. You see, the xylem and the phloem are what we call the fascicular band. We can call it band of sheath. We can call it band of sheath. Now, here we happen to have malic acid, and the malic acid is sending this carbon dioxide. It sends this carbon dioxide to the Kelvin cycle. Let's have a look at the Kelvin cycle and say it's the same as C3 cycle. I want to emphasize that. There, the carbon dioxide will be accepted by Lubib. You remember, Lubib, and then eventually series of reactions will occur which will generate for us an exhaust sugar. And the successful C4 plants would have formed the exhaust sugar like that. I want to emphasize that after discharge of this carbon dioxide, malic acid will form pyruvic acid. You see, it will form pyruvic acid, and the pyruvic acid will be discharged again to mesophyll cells, and the cycle will keep on happening. It is a significant difference that you should always realize. C3 plants differ from C4 plants this way. Because the C4 plants, they have Hutchinson pathway or the C4 cycle. It occurs in two groups of cells separated. While the C3 cycle occur, they occur only in the spongy mesophyll cells. The ability of this process to occur in two separate cells, I said we call it Kranz's anatomy. Kranz's anatomy. That. So they'll ask you what is meant by Kranz's anatomy, and you will explain the significance of Kranz's anatomy. It provides the enzymes that catalyze this reaction from different cells. It makes this process a bit more rapid because of the variety of enzymes provided by different groups of cells. If this reaction was happening in only a given cell, the rate of reaction will be slightly low because we shall have a single group of enzymes catalyzing the reaction. And enzymes can get exhausted if they are a few. It is the property of enzymes that they will increase. The rate of reaction will normally increase whenever enzymatic concentration is increasing. Now, if we have a constant concentration of enzymes here, we're going to expect reaction to be slightly low in C3 plants if compared to the Kranz's anatomy. There we have several cells involved with the different enzymes faster. So that's C4 plants. Students have a tendency of confusing these two statements. Students normally confuse the C4 plants and the C4 cycle. Just like they confuse the C3 plants and the the question sometimes technically asks students to give differences between C4 plants and C3 plants. So they ask students to give C4 plants, how they differ from the C3 plants. I suspect that the differences you're going to give here, they are slightly different from the differences you will give if they ask you to differentiate between C4 cycle and then C3 cycle. They're going to technically be asking you to give these differences because they're very many. Having appreciated the C4 cycle and the C3 cycle, you must be producing differences. However, I want to say, when we say C4 plants, we mean those plants that have both the C3 cycle in them and you also have the C4 cycle in them. Those are C4. So that's the plants are said to be C4 plants. They have both the C3 cycle in them and they also have the C4 cycle in them. I said that the C4 cycle, this is what we call a C4 cycle. The C4 cycle. We are writing that it occurs in the C4 plants. Within this cycle, we don't have any sugar formed. You can bear notice there. We do not form final sugar molecules. Even when malic acid and oxalic acid, we appreciate their carbohydrates, but the final sugar molecule we expecting that is utilizable in plants is glucose. And we will not produce it here in this cycle. Which it means that this plant of this nature, it must have a C3 cycle, which is predominantly known in formation of the sugar. I want to take it here briefly to remind you that if we discharge carbon dioxide here, this carbon dioxide will be accepted by carbon dioxide acceptor Rubico, 5 carbon compound, and this carbon dioxide accepted, this is carbon dioxide and is accepted. Time involved here, we said that it is Rubico carboxylase. Now, we have Rubic, it accepts carbon dioxide, it forms 6 carbon compounds. We mentioned that it is unstable. 6 carbon compound disintegrates to produce 2 molecules of phosphoglyceric acid. These are 3 carbon compounds. 3 carbon compounds each, there are 2 molecules. So that is understandable. And now we shall have this reduced to phosphoglyceraldehyde. We mentioned that there is a reduction reaction that exists here by addition of hydrogen with the NADP. This is the proton carrier, which is eventually discharged out as the NADP. That the reaction happens to be active. An ATP molecule is used to produce ADP and an inorganic phosphate. So we emphasize that condensation reaction will proceed here. This is condensation reaction, and we shall produce this sugar. Because sugar will be produced here, and then this is used to regenerate the Rubic like that. However, this reaction is active. An ATP molecule is broken down to ADP and an inorganic phosphate. Whenever an ATP molecule is broken, there is going to be some mark of emphasizing that the enzyme involved is said to be ATPase. This enzyme is usually utilizing catalyzing the reaction at which ATP molecule is broken down to ADP and inorganic phosphates. So now, have you seen that these C4 plants, they practice this cycle to only accept carbon dioxide even when it's at low partial pressures, and eventually they will fix this carbon dioxide at this stage in the Barlow-Sheet cells into the C3 cycle. This is the C3 cycle. So these plants have both the C3 cycle and the C4 cycle in them. Those ones called them C4 plants. So if they happen to ask you to give the differences between the C3 cycle and C4 cycle, don't you say that the C4 cycle carbon dioxide is fixed twice. Now that would be a difference if you were differentiating. It would be a correct difference if you were differentiating between a C4 plant and a C3 plant. It's where you would correctly say that the C4 plants fix their carbon dioxide twice. And the first fixation is by the PEP, accepted, and then the second fixation, carbon dioxide, is accepted by the Rubin. And then the C3 plants, for them, they fix carbon dioxide once. So I want you to sensitively give a question, give answers to a question when you're very careful to ensure that the answers you give are relevant or a correct response to that particular question when you ask it. So I want us to briefly talk about these differences, considering the two cycles, or considering the plants present here. The differences between the C3 cycle and the C4 cycle. I want us to record it there. You would probably construct a table whereby one side we have C3, C3 cycle, and the other side you have C4 cycle. Should use here. We have a table, and then we have C3 cycle, C4 cycle. All the students must be in position to generate their own differences here. So whenever they ask you about differences, it is correct that you present your work on your table, other than writing the while whereas, like the P7 students or pupils. Now, what you have here, you can see this, that the C3 cycle, carbon dioxide is fixed. Carbon dioxide is accepted by all people. C3 cycle, carbon dioxide is accepted by all people. Note that here. And this side of the C4, carbon dioxide acceptor is a PEP. Carbon dioxide acceptor is PEP. So the acceptor is the Ruby pool here. Now this is carbon dioxide acceptor. Now carbon dioxide acceptor is PEP like that. That's a difference. See? It's a difference. So you can go ahead and talk about the enzyme present here and the enzyme present here. So these will be self-generated. So the enzyme involved here is rubic carboxylase. The enzyme involved in this cycle is the PEP carboxylase. Those are clear differences. So PEP carboxylase is present here. This is the rubic carboxylase. That look around and see other differences. You realize that here, reduction of the sugars occur. Here in this cycle, we don't expect any reduction. We have not seen any NADH H2 used anywhere. So probably this reaction is reduction. Reduction occurs. Maybe reduction of sugars occurs to produce new products, and here simply say no reduction. That differences you can think of exhaust sugar is formed. Sugars of 6 carbon compounds are formed in this cycle, C3 cycle, we are forming these sugars. 6 carbon compounds, 6 carbon compound sugars form D. Come to this side, you realize that a maximum number of sugars formed only with 4 carbon compounds. That is oxaloacetic acid. Yeah, that is it. You remember Cranz's anatomy? Reactions occur only in one cell. The reactions proceed in two cells. In two cells, occur in two cells. That is what we call Cranz's anatomy. So you can simply say Cranz's anatomy occurs here. Cranz's anatomy occurs here, and this side we don't have any Cranz's anatomy. So probably that is what we have in the cycles and how these cycles differ. You can generate several others. Like for example, the intermediate carbon atoms formed in the C4 cycle have 4 carbon atoms. But the intermediate carbon atoms, the intermediate substances formed here, they have C3 carbon atoms. You can see the phosphoglyceric acid, phosphoglyceraldehyde, all of them have C3 carbon atom. But when you come here, this is presenting 4, and oxaloacetic acid is presented as 4. Those will be differences there. And among others. Maybe the very important one, very more last one, I want you to take note that the rubibucarboxylase enzyme for this C3 cycle, it operates at a relatively high optimum temperature. The enzyme is here, one of the temperature is one of the factors affecting them. This is operating, rubibucarboxylase operates at relatively the optimum temperature. Optimum temperature is relatively lower. So by 27 degrees Celsius, for them that is the maximum temperature. Any temperatures beyond this, they will be burning the enzyme. Unlike the pepcarboxylase, for them they relatively have a high optimum temperature that should be noted in the differences among the cycles. Optimum temperature here expected to be very high. Right there, you realize that the temperature here is going up to 45 degrees Celsius when this enzyme is still operating. That's a very good advantage there. Now, meet you in the other session, then we look at